It's great to have Exicure from Skokie here. Never seen a biotech company from Skokie.
Yeah, it's Chicago, so it's...
Wow, Skokie is nice. Sounds better.
It sounds better, yeah.
One of the broadest pipelines in biotech, I guess. Tell us about it, David.
Great, thank you very much. Pleasure to be here and presenting on behalf of the Exicure team all the way from Skokie, Chicago, Illinois. It's anybody watching the live stream, I think it's about 6 o'clock in the morning, so maybe somebody's at work. If they are, they get bonus points. But for everybody in the room, thank you all for being here today. Happy to tell you about Exicure. The stock ticker for those that are interested is XCUR, or you could follow us on our new Twitter account, execuretx.com. A little bit what I won't and will do today, so I will not be here with the purpose of selling stock, and of course I will make forward-looking statements, and what I really hope to tell you is a really important story about how we've solved one of the fundamental challenges for nucleic acid therapeutics, which is effectively delivery into the cells and tissues of interest, and how we develop that into a pipeline of drugs that we think are going to be compelling for changing the way we think about making and designing medicines. So a little bit about Exicure. Exicure originated out of Northwestern University just outside Chicago, actually based upon my PhD work with a gentleman named Chad Mirkin. The R&D facility is located in Chicago. We have about 30 folks in the laboratory, about 35 total people in the company. We are publicly traded as of May of this last year, and we've taken 3 drugs into clinical trial, 2 in dermatology and 1 in oncology. Our last raise was done in August of last year. We raised $22 million in a PIPE while OTC trading. Brings us cash on hand into January of 2020, so Q1 of 2020. In terms of the team that's been put together, we've developed a fantastic management team at Exicure. In addition to myself, David Snyder, the Chief Financial Officer, experienced CFO on the public side, Matthias Schroff from a company over here, who was previously at a company called Moligen and Vaxum as well, and Jocelyn Trokenheim, who joined us about a year ago from Takeda to head up our BD. On the board side, we have Chad Mirkin, one of the founders and a luminary in the field of nanotechnology, Shad Thaxton, our MD founder, David Walt, the founder of Illumina, Helen Kim, who spent the majority of her career at Chiron, but most recently was at Kite before she negotiated the transaction between Kite and Gilead, and Jay Venkatesan, who is a financial expert and spent his time at Brookside and Alpine. So a really fantastic team that's been put together to take Exicure forward. So what I want to chat about today is what we're doing at Exicure with our platform technology called Spherical Nucleic Acid, or SNA. And spherical nucleic acid is a platform technology that has many different applications, and as was mentioned, has built us a broad pipeline in the nucleic acid development space. In particular, we've shown that our technology can work in the skin, the eye, the lung, the GI tract, and even in oncology. And snapshots of all of those programs I hope to share with you in the next few minutes. A lead asset that we just brought into clinical trial is called AST-008. That's a TLR9 agonist that's being injected locally into tumors in patients with different types of tumors that are palpable. This morning, we're announcing first patient, first dose in the US in a phase 1B trial. So that'll be a Merkel cell carcinoma patient that is enrolling in this trial, and excited to tell you more about that here in a bit. In the inflammatory disease space, we've shown that local topical delivery of our SNA platform works in the skin. And in fact, just a few weeks ago, we announced our top-line results for application in psoriasis patients. And we also announced last week a partnership with a company called Dermalix. We are developing now a drug for Netherton's disease, a rare orphan disease based upon monogenetic skin mutations. And then finally, we've been excited over the last year to build out a part of the vertical in neurology. So developing spherical nucleic acids for local injection into the spinal cord or brain for applications in neuromuscular diseases. So with that preview of what's to come, let me tell you about what a spherical nucleic acid is. So an SNA is essentially 2 components that are arranged in a very specific architecture. So it's the DNA that you know and love here on slide 6, but it's arranged around the outside of a nanoparticle core. And why we think this is very different from what has historically been done in the nucleic acid field is that rather than encapsulate the nucleic acid inside of a lipid or polymer, we very deliberately put the DNA or RNA on the outside. So counterintuitive, and one would think that this is not the best strategy for hiding or encapsulating things for the purposes of getting across those very difficult to enter cell barriers. But what it turns out was a fortuitous discovery was that when you create these structures, they get into cells and tissues naturally. And so what you're looking at on slide 7 here is the naked DNA on the left-hand side, and that same DNA, the same number of strands of DNA, just arranged in 3 dimensions to form the SNA on the right-hand side. So if there's one lesson today scientifically, that's a takeaway. Architecture matters. 2D architectures of DNA don't get into the cells. Once you start looking at the world in 3D, as we do with our spherical nucleic acid structure, you can see that the cells light up, and now we've got effective delivery into the cells of interest with the 3D structure of the DNA or RNA. We know quite a bit about how this is done now biologically. In 2013, there was a paper that came out of Northwestern University in PNAS that showed how they get in through a scavenger receptor mechanism. And in essence, what's happening is a multivalent binding, or multiple bindings are happening between the DNA and these scavenger receptors at the same time, which triggers an endocytotic process and brings the nanoparticles into the cells. Why this is cool is a natural process. It's an active process, and the spherical nucleic acid we find goes into all cells and tissue types that we look at as a result of this scavenger receptor expression, which seems to be ubiquitous across various different cell types and tissue types that we've looked at. As a result, we have been one of the first companies to take nucleic acids outside of the liver space. And so the idea here is, can we expand the opportunity set for nucleic acid therapeutics and going after different opportunities biologically now that we've cracked the delivery challenge? Far more than a science project or just a delivery technology, we've actually developed drugs on this platform now. And if you look at slide that's on the screen, what you can see here is that we've developed across multiple different therapeutic areas. And in particular, we have 2 programs in dermatology that have advanced through phase 1 completion, and 1 program in oncology, which completed a phase 1 trial in the UK, and is currently undergoing a phase 1b/2 study in the US. We've also started and declared a program in neurology. We're expecting to nominate a clinical candidate before year end for neurology. And ophtho, GR, and pulmonary are areas I won't have time to talk about today, but are areas where we're actively seeking partnerships based upon the preclinical data packages that we've collected. So first vertical I'll talk about is the immuno-oncology bit. And here the idea, if you're not familiar, is that one can use so-called checkpoint inhibitors as an immunotherapy. The checkpoint inhibitors take the brakes off the immune system. Great year for checkpoint inhibitors last year in terms of approvals, but also in terms of recognition. The 2018 Nobel Prize in Medicine actually went to the developers of checkpoint inhibitors. But we can do better when it comes to checkpoint inhibitors. So taking the brakes off the immune system is only one aspect of it. You also want to hit your immune system with a high octane gas. And so if you can take so-called cold tumors and make them hot by both releasing the brakes off the system and hitting the high octane gas, we think that 1-2 punch is really going to be transformational when it comes from taking those 30 or 40% response rates for the checkpoint inhibitors much higher for future generations and combination therapies. Last year was also a great year for the strategy for activating the innate immune system. In particular, TLR9s were successful with readouts from Dynavax, Checkmate, and IDERA. All colleagues and competitors in the space, which showed that this premise of activating the immune system through toll-like receptor pathways in combination with checkpoint inhibitors was a successful strategy. So why we're so excited about Exicure's platform is on slide 13. And that comes back to that issue that I showed you in the front, which is the delivery has always been a challenge for nucleic acids. And if you can deliver more into cells and tissues, you can have higher potency. So on slide 13, what you're looking at here is the separation in a red line of a linear nucleic acid structure, this being a historically used Pfizer Coley CpG 7909 sequence. And the blue line is when we take that same sequence and arrange it into 3 dimensions to create the SNA architecture. And so simply by going from a 2D to a 3D architecture here in the animal models, we see again that the potency goes up by about 100x. This type of paradigm was successful preclinically in breast, lung, melanoma, colorectal, and bladder cancer, and was successful intratumorally injected, subcutaneously injected, and even IV injected. So high potency TLR9s as enabled by the 3D structure. So where can we take this strategy of activating the immune response? Well, we're going after large tumor populations like melanoma, head and neck, and squamous cell carcinoma, but we're also going after some places where people have not yet tickled the immune system in combination with these checkpoint inhibitors. For example, Merkel cell carcinoma. So Merkel cell carcinoma, for those that don't know, in the US is about a 2,500 year case patient load. we think it's an opportunity to improve the current standard of care checkpoint inhibitors with a combination trial like our AST-008. And that's one of the trial sites that we have set up and running, and one of the patient populations that we're targeting for this phase 1B approach. Top line data for the phase 1B will be completed by year end. So we'll have something to say about the possible 18 patients that will be enrolled in this trial. Okay, so the next part of the platform is the genetic disorders. And here, think about the DNA or RNA as targeting mutations. So places where genes have a mismatch or other type of deletion or gain of function mutation. And can we use now DNA to go into those cell and tissue types and correct those? Our test case here, which I think is really compelling, is spinal muscular atrophy. And the reason spinal muscular atrophy was of interest to us is, for those of you that aren't familiar, Biogen had a great success over the last year and a half with the approval of Spinraza, also known as nusinersen. And so Spinraza is currently being used in patients and kids with spinal muscular atrophy. But what we found is that if you take the spherical nucleic acid format of the Spinraza, that you get much better potency, again, by creating the 3D architecture. So the exemplar slide here on 16 is the red line is a single dose injection in the mice of Spinraza, nusinersen. And when you take that 3D format of it, you see what happens in the blue. So same nucleic acid again, same drug developed by Biogen Ionis. You put that in 3 dimensions, and now the potency goes up about 30x. In vitro survival goes up 4-fold simply by creating the 3D architecture. So hopefully I convinced you again that those architectures are really important, not just for drugs that we're developing internally at Exicure, but also ones where people have good nucleic acids that are being developed clinically and being clinically applied to patients in need. We think we can make them excellent by creating 3D formats. Perhaps one of the most exciting pieces of data for us last year scientifically was this slide shown on slide 17, which is that when we create these 3D structures, not only do we get into the cells better, but we also stay within the tissues of interest much more avidly. So what you're looking at on this slide is a comparison at 3 time points, 0, 24, and 7 days out, 168 hours of nusinersen in the 3D format of the nusinersen. So what you see here is that single dose injection, again at time equals 0, same number of nucleic acids go into rat. By 24 hours, what you see in the case of nusinersen, that middle column, is that it starts to leak out in kidney, liver, spleen. In contrast, if you look at the spherical nucleic acid format, what you'll see there is that it stays inside the brain and the spinal cord. Rolling that tape all the way forward out to 7 days, what you see the importance of that, it remains throughout the brain and the spinal cord. So better potency is driven not only by better cellular uptake, but also better penetration into the tissues of interest, in this case, the brain and the spinal cord. So on the neuro side, compelled by this data, we've looked at spinal muscular atrophy as an opportunity. We've also been looking at other diseases. We're expecting to nominate candidates in the neuro space by the end of the year. Options that we're currently looking at include Friedreich's ataxia, Batten's disease, spinal bular ataxias, and others. So we'll have more to say about that here in 2019. So the final bit that I'll talk about today is our inflammatory disease pathway. And the idea here is think about those nucleic acids around the outside as ones that are hitting inflammatory pathways. So maybe places that you know and love from the antibody space. TNF-alphas, IL-17, interleukin type pathways. And if we design the DNA to go after those pro-inflammatory pathways, we can tamp down those inflammatory pathways and go after, instead of systemic drugs, go after local opportunities hitting those exact same biological pathways. So the case study here for us has been Exicure 17. Exicure 17 is a locally, topically applied antisense molecule directed against the IL-17 receptor. And what you can see on this slide in the image is a picture of a patient that has a psoriatic plaque. When they have the psoriatic plaque, we have the opportunity to put 5 different application sites on a single patient. So low, medium, and high dose of our spherical nucleic acid, and then a positive and negative comparator. Negative comparator being the gel only, and the positive comparator being the currently used Dovanex, clinically used vitamin D analog. At the end of a 26-day trial, we were excited to report here recently that 20 of the 21 patients treated, 11 showed a clinical response at the high dose. Now, this for us was somewhat unexpected for this very short trial duration. We're already seeing efficacy with a local topical application of an anti-inflammatory targeted at the gene of interest, the IL-17. So we're very excited about this data. Again, it's, I think, the first time in our knowledge that anybody's demonstrated that local topical gene regulation can lead to a clinical effect in patients. And on the heels of that, we were excited to announce last week a partnership with a company called Dermalix. Dermalix is developing rare orphan skin disease drugs, and they are going to be working with Exicure jointly first on a program in Netherton syndrome. So I encourage you to take a look at that. Just a few things to sum up here in the remaining minute. Exicure, we think, is unique in that we're not defining ourselves based upon the biology in which we target. So we work across the different nucleic acid modalities, the siRNA, the antisense, the toll-like receptor spaces, the splice correction like the Spinraza, etc. And all of that allows us to not only access different targets and organs of interest, but to access them through different mechanisms. So in contrast to competitors in the space that narrowly focus on siRNA or antisense or one particular biological modality, I leave you with the idea that spherical nucleic acids can work across all those different modalities, and we think provides a compelling opportunity for expanded delivery options in different tissues of interest. So in summary, lots to watch for at Exicure this year. We have this phase 1b study that is currently open. It's open at Dana-Farber, John Wayne Cancer Institute, University of Miami, as well as University of Iowa. We'll be enrolling patients and reporting out top-line results before the end of the year. We have our Exicure 17 results, so more to talk about on our anti-inflammatory pathway in the year to come. We were excited to see multiple patients, over half that were treated, show response in the clinical trial. We think that's the tip of the iceberg when it comes to dermatological disease. For example, there are about 200 or so that we think are applicable with either inflammatory pathways or that have genetic basis. And then finally, the expansion of the neurology space. So we have this tremendous opportunity with the data and knowledge in hand from the nucleic acid space and from the gene sequencing to go after opportunities where genetic drivers of disease in the brain, for example, are places that now we can access with a technology like spherical nucleic acid. So with that, I'm happy to give you my contact information, davidg@exicuretx.com. And for those following along at home, thank you very much for your attention, and happy to meet anybody after the session here in the room for a private chat. Thank you.